Alright, so here we are back at the enzyme, and we're looking at an enzyme here on the left where we see in here this area we're calling the active site, and within that active site now we can see the substrates of this particular enzyme. So this is showing that idea of an induced fit, so that the substrates fit into these areas of the active site, and then the enzyme actually kind of snuggles around and puts some tension on these substrates to help with the process of either catabolism or anabolism, depending on what's the product. So let's take now a look at one other thing, or actually a couple. But so sometimes enzymes need partners, and these are some examples of things which are non-proteins, but they are necessary for certain enzymes to do their role in catalysis. So here is the type of molecule we're talking about, and here is kind of its role when it's working with an enzyme. Now this part in here, not important that you know, at least we'll talk about some more of these coming up. But this you can see some of the differences. So cofactors are things that you might be able to see directly on the periodic table, things like iron and copper and zinc. So just elements, atoms of certain elements which are combine with enzymes, and then they can help them to work. There are things called coenzymes. Now you may notice coenzymes have things like biotin, so you've seen that before, or coenzyme A. So some of these are actually what we talk about as vitamins here, but sometimes a coenzyme might be ATP itself. Then there can be things called prosthetic groups. Now the one that you probably are familiar with is heme, because this prosthetic group is a part of hemoglobin. So this is it's an iron-based compound that's a part of, it partners with some enzymes. So just know that some enzymes require non-protein components to them in order to help them to work. So that's kind of the take-home here. We looked at this on the POGL, understanding that the reaction rate of an enzyme can be measured in a variety of ways. So we can see that on this graph it's looking at, there's a reaction, one which has an enzyme, and here's a reaction that doesn't have the enzyme. And you can see there's an increase in the concentration of substrate here on the x-axis, and that the reaction without the enzyme is still taking place. It's just at a much lower rate. So it would take much longer to get whatever product we wanted. So the benefit of the enzyme is we can speed up that reaction until it hits this maximum rate. And at this point, on this flat part of the line, then this reaction, in this case, the enzymes that are doing this are saturated. So they all have substrate in them. They can't go any faster than that. They can keep dropping off the product and picking up more substrate. But this is just saying this would be the maximum activity rate of those set of enzymes for no matter what the concentration is. So as you go higher in concentration, it doesn't matter because they can't go any faster. That's kind of that Uber driver thing that I mentioned. So this is basically looking at the same thing. And so when substrate concentration is very low, we're going to have a fairly low rate of activity here. And as you get more substrate, then you're going to have faster and faster activity. So that kind of makes sense. We also know that the environment can change the way that enzymes operate. So changes in pH. Although in this graph, if you look at it, it's telling us what the optimum pH is for a particular enzyme. And we've looked at pepsin already. We know that this would be one associated with the stomach. Okay, so protein digestion kind of starts here. And then over here we have something called chymotrypsin. And remember, we looked at trypsin on the POGL. This is a particular type of trypsin that works on certain amino acids better than others. So this is also a protein, and this will break down proteins. In particular, I think it kind of works on the aromatic types of amino acids. And then arginase is one that's more associated with getting rid of waste products. So this is one of the last steps in forming something called urea, which becomes urine, which is what we get rid of there. But all three of these have an ideal pH under which they operate, and you can kind of see what those are here. Also temperature. So most enzymes operate at body temperature, so they have sort of an optimal temperature. And if you decrease the temperature, then it's going to slow down that rate. If you increase it, it's also going to slow down that rate. And to the point where if it's extreme here, or if it's extreme in the pH for that enzyme, it's going to start to denature it, and therefore it's not going to work as well. It'll seriously slow it down. So we're just getting a bigger view of it here. So the environment can change the activity rate of the enzyme. So most enzymes, therefore, by design, are designed to operate in the range that's ideal for them. So pepsin's found in the stomach because that's what the pH of the stomach is. Chymotrypsin and trypsin are found in the intestinal tract, so that we see the pH is different. And then the arginase is, again, found in a different location. This would be more associated with our kidneys and our urinary tract. Okay, so I think that's sufficient on that. But there are other ways that enzymes can be influenced. Their activity rate can be changed. One of them is this. So this is DIPF. It's just an example of actually a neurotoxin. But this is what we'd call an inhibitor. It can go in to the active site and can bind to that active site so that this enzyme, which would normally break down something called acetylcholine, can't do it. And not only can't it do it, it can't do it ever, because once this is bound, it's irreversible. So this would be a toxin that would be a deadly toxin, because once it binds in this enzyme, the enzymes can't work anymore. So that's not a good thing. But there are lots of ways that inhibition is not a bad thing, sometimes is a necessary thing. And what we're seeing here is that sometimes there is an inhibitor that can go into the active site, and when it's there, this enzyme cannot be active. And when it's, so you can see here the substrate can't go in. So that's called competitive inhibition. So something is competing with the normal substrate for that active site. So that's competitive inhibition. And competitive inhibition is reversible. So this can go in here, it can bind, but then it's going to leave, and so it's going to slow down the rate of this enzyme. A non-competitive inhibitor is something that doesn't go into the active site, but what it does is it goes into a different site on that enzyme, and when it does, it changes the shape. So if it changes the shape of the enzyme, so this isn't denaturing, but in a sense it looks like that, because now the enzyme's shape has changed, therefore but it's changed because of this. It's sort of more of a mechanical action that the substrate can't fit in. So that's non-competitive, because the active site is still available. In other words, nothing is blocking it, but this is changing the shape of the enzyme, so now the substrate can't go in. Okay. So again, here's just kind of the comparison. Competitive, non-competitive. So this picture is not big enough for me. There's a little bit bigger one coming up here, but I'll leave this up here just to show you because this has some little, little descriptions along the way, what's happening. So here's an inactive enzyme, and it is just kind of hanging out, waiting for something to activate it. So sometimes something needs the enzyme, instead of being active, it needs something to activate it. So this is an example of when something can go into an area that is not the active site, so much like what we just looked at in this non-competitive inhibition, but this is not inhibiting, this is activating. So when this comes in and it changes the shape, so notice here's the activator, comes in, and that actually opens up the active site. That's called an allosteric or change of shape, or steric refers to shape, and it's going to bind, opens up the enzyme's active site. Now the substrate can come in. Now the product is released, and now, whoops, and there we are on that side. So we have to go two ways. So that's one thing that can happen. Another way that can happen too is that an inactive enzyme can be activated by putting a phosphate on it. So here what you see is this is being phosphorylated. Just means you're adding a phosphate to it, and that by itself will activate this enzyme, and it'll produce the product. So let's look one more time here, a little bit clearer. Two ways that we can change the shape of the enzyme in a way that's going to, in this case, open up that active site so that it can do its job. So this is allosteric regulation. It can happen because some molecule that acts as an activator can go in and open it up, and then it can create the product because the substrate can go into the active site. Or we put phosphate on it, phosphorylating it, and here's the thing that's actually going to phosphorylate it, and here it is, and now we've got the product. And so when these things happen, you don't need to memorize this at all. We're going to be looking at a number of these biochemical reactions, but what this is showing you is that most metabolic reactions, most things in our guts, our metabolic pathways, are many steps. They start with something, that gets converted with an enzyme to something else, and then every single one of these little green arrows is representing another enzymatic reaction. So another enzyme's involved here, changes to something else, to something else, to something else, to something else. So you finally get to this product. So this is the desirable product here at the end, this thing called isoleucine. But then what happens is, if we do this over and over and we get a lot of isoleucine, now isoleucine can feed back on that very first enzyme. Some of this end product will go back and shut this off. And this is called feedback inhibition. You don't want to make more than you need, so once you have enough, you want to turn off the faucet. And so some of the product itself is used to inhibit that first step of the reaction. So in other words, whatever enzyme is active here is now going to get inhibited. And then finally, not to get you too excited, but this last part right here is looking at what's coming up. And so here we see glucose, our friend, and we're going to take glucose, or our cells will, and they're going to change it, they're going to convert it. And they're going to have multi-steps to do that. So we can see here every one of these, 1, 2, all the way down, there's 10 steps here where an enzyme is involved, changing glucose to some intermediate product, to something else, and continues to do that until we get to something at the end. So this is actually the preview of something called glycolysis. So we're going to take a look at glycolysis in the cell, which is one of the most important things we can talk about in biology. So stay tuned, and that's our last thing. So there you go.